Cells have an orderly structure. They respond to external stimuli, they regulate their internal environment, they grow and they develop, they process and utilize energy, and they reproduce and evolve. So we're going to talk about cellular reproduction now. There are two kinds of cell division that we're going to talk about in this class, but in Unit 2 we're only concerned with one, and that's called mitosis. Mitosis is the form of cell division in which a single parent cell divides to create two daughter cells. The products of cell division are referred to as daughter cells, and those two daughter cells will be genetically identical to each other. Not only to each other, actually, not only are they twin daughter cells, but they're also genetically identical to the mother cell. The other form of cell division, which we will attend to much later in the course, is called meiosis. And in that form of cell division, you actually end up with 4 daughter cells, which are genetically unique from each other, and genetically distinct from the mother cell as well. Another difference between mitosis and meiosis is that in mitosis, you end up with the same amount of genetic material in the mother cell as you do in the 2 daughter cells at the end. So you might be detecting that mitosis is conceptually a little bit easier to get a handle on first. So that's why we're going to start out with mitosis, and then later we'll deal with meiosis. The basic process of mitosis is this. All the while a cell is doing the ordinary business of being a cell, it is constantly in a process of duplicating the genetic material. It's constantly taking all of the DNA and it's copying it. It's making a backup file, if you will. It's making a second copy of all of the genes. And when it's ready to divide, it's going to take all of that DNA that's in the nucleus, all of that chromatin. Remember, I called it spaghetti on a plate. It's just one big globular mass. You can't really see individual chromosomes. When it's ready to divide, we start bundling up those individual chromosomes. So suddenly you can see individual chromosomes. You can see individual package, individual noodles of DNA. And we're going to separate them out. But in order to separate them, we need to get rid of the nuclear envelope because it's kind of in our way. So we break that down. And then we have 2 copies of every chromosome because we made a backup file, right? So we separate those from each other, and we put one copy over here on one side of the cell and another copy over here on the other side of the cell. So now each side of the cell has one full copy of all the genetic instructions for that organism, and then we pinch the cell in half so that you have 2 cells now with a complete copy of the genetic instructions, and then those 2 clusters of DNA reform into 2 new nuclei, and we have 2 daughter cells which are genetically identical to each other and identical to the original mother cell. And they then begin the process of putting themselves back together, growing back to their original size, developing back into mature cells. And when they're ready, they begin doubling their DNA as well. And when they have a backup copy of their own DNA, they get ready to divide in half again. Now, that's the basic set of processes, but you need to learn the phases of mitosis, the step-by-step process in which this occurs. One question that's good to keep in mind, because it's always important to stand back for a moment and think, wait, why are cells doing this at all? Is why do they do mitosis? Why do cells divide? Well, there's a couple of reasons. One, as I've said before, nothing lasts forever. No system can run eternally. So every now and then, cells are going to age, they're going to die, they're going to scrape off because you got an abrasion or something, you scraped your knee, something like that. So you're going to have to replace old cells, or you're maybe going to have to reproduce. Maybe a single-celled organism needs to replace itself. Or maybe you are a child and you want to grow up to be an adult. Well, you don't grow by having your cells get bigger. You grow by making more and more and more cells. So there's lots of reasons why your cells might need to undergo mitosis. And again, mitosis is the form of cell division where 2 daughter cells are produced that are genetically identical to each other and to the original parent cell or to the original mother cell. Most cells spend a relatively tiny amount of time in mitosis, though. It represents about 10% of their total life cycle, and they spend the rest of their time in a phase which we call interphase. Interphase is just the cell being a cell. Not, it's 90% of its life. It's when it does all of the normal functions of being a cell. But even when it's in interphase, in the background, it's always planning for the next mitosis to take place. So we'll talk about what it's doing in terms of planning for cell division during interphase, and we'll talk about what it's doing during mitosis as well. In eukaryotic cells, all of the genetic information for the organism is contained in the DNA, in the chromatin, in what are called genes. Genes are just sections of the DNA, sections of the chromosomes, which code for one particular protein, and of course proteins do all of the work inside of your cells, so if you have a complete set of the genetic instructions for making all of the different proteins, you have a complete set of the instructions for putting together the organism. Genes are located on the chromosomes, and the chromosomes are of course located inside the nucleus. Before a parent cell splits in two, it's got to duplicate its chromosomes. Okay, so it's going to go from having a single copy of every gene to having two copies of every gene. And it goes from essentially having one leg of every chromosome to having two legs of every chromosome. Now, the cell cycle describes an oscillation between these two states of being that I just mentioned, interphase and mitosis. So it goes through the metabolic activities of life and suddenly switching gears entirely to going 100% into cell division. When a cell is getting ready to divide, everything in the cell has to be shifted over into cell division mode. There's no time or no resources left to be doing any other kind of cellular functions inside the cell. Everything is committed to mitosis when it's mitosis time, which is part of the reason why mitosis is such a short segment of the cell's life cycle, because if it was the majority of what the cell was doing, you wouldn't be able to do much of anything else as an organism. All your cells would be dividing all the time. They wouldn't have time to keep the rest of your physiology going. Each daughter cell is going to receive one set of the chromosomes, so that's why it has to duplicate the genetic information first. So that phase in which the cell spends most of its time is referred to as interphase. And interphase is divided into three subphases. The first one is called G1. G1 is followed by S phase, and S phase is followed by G2 phase. Now, at all points of interphase, the cell is going about the normal business of being a cell, but in different parts of interphase, it's making different preparations for the next mitosis. G1 is gap phase 1. That's what the G stands for, is gap phase, but I like to think of it as growth phase 1 because that's primarily what's happening. The cell is growing during gap phase 1. It just finished its last mitosis, so we've just split into two daughter cells and they are now half the size of the original parent cell. So what do they have to do? They have to put on some size. They need to recover from that cell division. We're going to need to prepare for the next chromosome doubling. We're going to need to synthesize more ATP. We're going to need to make some more nucleotides. We're going to have to regenerate some of our organelles, make some more endoplasmic reticulum, make some more membranes. We're going to need to make more enzymes and just generally prepare to get back into tip-top shape. Once gap phase 1 is completed, then we enter S phase, and S stands for synthesis. To synthesize is to make, so we know we're going to make something in S phase. What are we going to make? We're going to make some DNA. S phase is when the DNA is replicated, when we make two copies of every gene. So every chromosome goes from having one leg to having two legs, which you will see. Every chromosome gets a second chromatid, is what it's called. You go from having a single chromatid, one stick of information, to having two sticks of information attached to every single one of those packages. So you get two copies of every single gene. So you have twice as much genetic information as any cell would actually need in order to go about its normal business. So S phase is the DNA synthesis phase. Now, does this mean that there are a total of 2 of each kind of gene? No, actually, it means that there's 4 of each kind of gene now, because we are what's called diploid organisms. Every chromosome that you get from dad, you also get one of those chromosomes from mom. So any gene you get from dad, you probably get one of those genes from mom, except for if you're male, you get an X chromosome from mom and a Y chromosome from dad. So there's a little bit of asymmetry in your chromosomes there. But for the most part, you already have 2 copies of every gene. So at this point, you actually have 4 copies. However, the versions you got from dad and the versions you got from mom can be different from each other. And after S phase, the copied versions are going to be identical. So you actually end up now with 2 pairs of chromosomes. You have 2 identical versions of your dad's chromosome, and you have 2 identical versions of your mom's chromosome. Okay, so you have 4 copies of the gene, but 2 pairs. All right, once we finished duplicating all of the genetic information so that we have twice as many chromosomes as any cell would rightly need, we enter gap phase 2, which is another round of growth, because we got to prepare for this next cell division, right? We got to get up to a big enough size that we can sustain splitting, rupturing ourselves in half, our cells in half, and becoming 2 daughter cells here. And cell division is going to be a process that requires a bucket load of energy. So we need to make a lot of ATP. We got to make sure that we have extra microtubules because it's going to take a lot of mechanical support to be able to wrench these 2 daughter cells apart from each other. It's going to take a lot of cytoskeleton work in order to make that happen. We're going to need to make sure there's enough cytosol to go into each of these daughter cells. We're going to need to make sure that there's enough organelles to end up in both locations, right? They both of them need to have enough of a good starting point to survive to get through the next growth phase, and then to go through synthesis, and then to G2, and then to get to their own next cell division. So G2 is a second round of growth in order to prepare for the next mitosis. All of this is inside of interphase, okay? G1, S, and G2, that's all interphase. And interphase is 90% of a cell's life. Now, some cells don't proceed through mitosis after a certain stage of development. So brain cells, for instance, they don't really go into mitosis after a while. So they would go into another phase, which we call G0, which is kind of a resting phase. They are still in interphase, and they're going through the process of being a cell, but they're no longer preparing for another cell division. They are just keeping it steady. They're holding it together, and they are waiting for a signal that tells them what to do next, and that signal may or may not ever come. But if they get the right signal, cells can then be initiated to begin mitosis. And then mitosis is the process of cell division. Now, there are other kinds of cells that can be pushed into a different kind of cell division called meiosis, which I mentioned before. That only happens in your sex cells, though. That only occurs when you're making sperm and egg cells. And right now, we are only concerned with mitosis, which is the formation of 2 diploid identical daughter cells. Diploid means that you have 2 copies of every chromosome. So that's what we're making. And this is how you replace any cell in your body, any body cell that you have. So if you scrape some skin cells and you need to heal, mitosis is how you're going to heal that. Now I'm going to talk about the stages of mitosis. One thing you're going to notice is that the majority of our attention in the stages of mitosis has to do with what's going on with the chromosomes at each point in time. And that's because the chromosomes contain all of the genetic information, all of the instructions for making all of the enzymes, all of the proteins, all of the structural and functional units of the cell. So it's the most important thing for each daughter cell to have. They can, be missing other types of structures inside. They can be missing a little bit of membrane. They can be short a little bit of ATP. They can be lacking a little bit of cytosol. But if they're missing a few genes, that could be cataclysmic to the development of that daughter cell. So the most important, the most vital thing that each daughter cell has to have is a complete set of genetic instructions in order for it to develop and become a healthy cell. And if we're talking about a developing organism, like going from a single cell up to a multicellular adult, then losing any of those genetic instructions at an early stage would be horrible for the adult organism, because you would lose vital genes in every cell in that organism. So we have to make sure that we are going to sort out these chromosomes properly. In Interphase, we have the chromosomes in the kind of spaghetti on a plate model. So this is actually a plant cell. You can tell because of the cell wall, the nice rigid structure we have around the outside. You can see the nucleus right here, and the nucleus has chromatin inside of it, and you can't see individual chromosomes inside of that nucleus, but you can see a nucleolus, can't you? Look at that, the little buddy inside the nucleus, and that's just a dense region of chromatin. It's the part that's associated with making ribosomes, parts for ribosomes. They're assembled in the cytoplasm. And during interphase, it's that spaghetti on a plate model. All of it's loosey-goosey and in a big old blob inside the nucleus. But once we enter mitosis, our first phase is called prophase, and you can identify prophase because the first thing that happens is that we condense the chromosomes. We wrap up each chromosome, each string of DNA, into one tight little package, and we break down the nuclear envelope. And because we wrap up the DNA into as tight a package as possible, no one region of chromatin is any more dense than the others, so effectively the nucleolus disappears. There's no more denser region than any other, so there is no nucleolus anymore. So chromosomes are made of chromatin, which is a combination of DNA and protein molecules. So the proteins are called histone proteins, and they're there to sort things out. Each long chromatin fiber is a chromosome. So each chromosome is one long string of DNA. And if we were to line them all up together, you'd have 6 feet of DNA inside each one of those cells. And individual chromosomes cannot be identified until mitosis begins. Once you're in mitosis, then you can say, there's a chromosome, there's a chromosome, there's a chromosome. But before that, it's just one big old blob. All right, the first phase of mitosis is prophase. I like to think of prophase as the packing phase of mitosis. This is my little mnemonic that I use, so the P in packing, the P in prophase, that helps me remember things. So we're going to take the DNA and we're going to wrap it up around these histone proteins here. We're going to wrap it up around them nice and tight into these little bead on a string conformations, which we call nucleosomes. That's what you get here, these nucleosomes. And then those nucleosomes are wrapped up even tighter into these helical fibers, and then those helical fibers get wrapped up even tighter into looped domains, which eventually get wrapped up even tighter into chromosomes. And if you take a look at down here, this is an actual transmission electron micrograph of a chromosome. You can see that it has 2 different legs, and I have a colorized scanning electron micrograph up here of some chromosomes as well, and you can see that they have 2 different legs. Each of these legs is referred to as a chromatid. These are sister chromatids. So remember when I said that one of the things that happen in interphase is that we're going to duplicate the chromosomes? Well, when that happens, we create this second leg, this second strand of DNA. So these are 2 strands of DNA that are identical to each other, that are joined at this narrow little waist, which we call the centromere. But each one of these chromatids have exactly identical genetic information on each of them. They're storing the exact same codes. If this one says blue eyes, the other one says blue eyes. If this one says short, the other one says short. Hairy back, okay? It'll say exactly the same thing on both sides. So the process of the cell cycle is really doubling the genetic material by creating sister chromatids, and then in mitosis, you break apart those sister chromatids to create daughter chromosomes. So during interphase, the cells duplicate their chromosomes, resulting in 2 copies of the genetic information called sister chromatids. Sister chromatids are joined together in a narrow waist in quotation marks, called the centromere. During mitosis, sister chromatids separate from each other and become daughter chromosomes. Once separated, each chromatid is at that point a fully fledged chromosome. We don't call them daughter chromatids once they're separated, they become daughter chromosomes. And they will be identical to that original chromosome. There's no shuffling of genes. In mitosis, we are just duplicating genetic information. Here it all is arranged like a big old clock. Let's start at the last cell division. Since it's a big circle, we can kind of start wherever we want. I think it makes the most sense to start at the last cell division. So we have a new daughter cell that is in G1 phase, gap phase 1, of interphase. And that means it's in a growth phase. So it's going to add more cytosol, it's going to generate more ATP, it's going to create some more organelles, it's going to regrow its membrane. And then once it gets up to a good size, it can get back to its normal functioning. It goes back to its job of being a normal cell. It does everything a cell's got to do. But the nucleus is going to go to work synthesizing more DNA. And this is when, if you look at the chromosome here, the picture of the chromosome, it's going to go from a single chromatid chromosome to a double chromatid chromosome. You have 46 chromosomes inside of your nucleus, so all 46 of them are going to go from single chromatid chromosomes to double chromatid chromosomes. And then, once that synthesis phase, that S phase has been completed, you're going to get into G2 phase of interphase, which is going to be another growth phase, gap phase 2. And once it gets big enough, once it gets so big that it really has no choice but to split in half, because it's so large, it has basically no other choice, then it enters the mitotic phase. It enters mitosis, which, since this video is already getting to about the 20-some minute mark, I think maybe I'll finish that up next time.